Welcome everyone. So let's begin, and while the latecomer will join, let's just begin. So is your cell therapeutic product free from genetic abnormalities? A question you should ask yourself, of course, if you develop such products. Today I want to welcome you and thank you for attending this webinar. So we will talk about Karyology, cytogenetic assays, to spot the genetic abnormalities into cell medicinal products. So let's get to know each other better. So myself, I'm Aymeric on the right here. You can see my picture. So I have an engineering background, and I've done clean cells since 1.5 years. And I have the mission to develop the cells and handle the project with the objective to put your drug in the market using, of course, clean cells capabilities. And today I will give the speech to Vasileios, our expert in cytogenetics, and I will let him present himself.
Well, good afternoon, everybody. I am Vasileios GEORGAKAKOS, and I am the cytogenetics manager at Clean Cells. I hold a PhD in cytogenetics, and following now almost a decade of research and published work in the cytogenetics of leukemia, I joined the Clean Cells team back in 2012. My principal tasks back then were the creation of the in-house cytogenetics laboratory, but also the development and validation of the Karyology and FISH assays in-house. Since then, we have completed hundreds of projects, and we will share some of our experience with you today. Back to you, Aymeric.
All right, thanks, Vasileios. So before we begin the actual presentation of the Karyology and such, I think it's important to give you a quick overview about Clean Cells, who we are, because we are not just a Karyology company. So what we do here at Clean Cells is that we will produce in GMP the cell line and the master seed stocks, viral seed stocks. We will offer the characterization of those banks, and we will also provide some external capabilities of quality control of the banks, of course, but also with the intermediate of production. So, for example, the bulk harvest, the end of production cells, and the drug substance, drug product. So everything the different pharmacopoeia ask to put your drug on the market. So we are able to work with various biological products, such as vaccines, antibodies, gene therapy products, cell therapy, and phages. We are located in France, so exactly near the city of Nantes city, in the Atlantic coast of France. And we have around 110 employees on site. The company was created in 2000, and each year we are able to conduct 1,200 studies, which represent approximately an average of 5,200 sample analyses. Less than a fifth of our workforce is dedicated to research and development, allowing us to stay on the forefront of innovation and to develop a customized assay for our clients. You can see here that mainly we work with of course, pharmaceutical companies, biotech companies, but also other CMO because they want to stay focused on their core activities, while we can offer a very renowned service in the industry for the production, like I said, of viral banks, of cell banks, and of all the associated quality control. We are mainly working in Europe here, so France, Europe, and also less than 15% in the US, and 7% in other countries. while you also have to keep in mind that our work in Europe are with other CMOs, like I said, and often they work themselves for US companies. So we are really global and working with much varieties of pharmacopoeia, whether it is US pharmacopoeia, European pharmacopoeia, and even Japan and Chinese, you name it. All right, so it's done with this overview of the company. So let's the presentation of Vasileios begin. Here is the agenda for this presentation. So he will do a short introduction about how he managed to implement this service into a full GMP compliant assay and services. He will also demonstrate how it works basically, and we will talk also about regulation, why you should do that and how you should do it. We will talk also about the recommended assays, and then we will share two interesting case studies, one about the CAR T cell product and one about a stem cell product. So at this point, I should disclose that those case studies are a representative study. They are not focused on one product of our clients in particular. They are much more like a representative study, and we give really the focus on the scientific aspect of the investigation. And I guess this is really the important part, which Vasileios will elaborate about. So during this conference, we want it to be as most interactive as possible. So you can ask questions using the right panel here. So you just have to click on the button here, questions, and then you can type your question. We will have two sessions of question and answer, one in the middle of the webinar and the other question at the end. So it's supposed to be a 30 minutes webinar, but at the end of it, for those who have questions, we will just take the time to answer. Okay, so Vasileios, I'll let you the microphone and get it done with a presentation and tell us everything there is to know about Karyology and cytogenetics.
Thank you very much, Aymeric, for the introduction and today's invitation, of course. We will have the opportunity to talk about our experience in addressing a critical question for any cell-based medicinal product manufacturer. Is your cell product free from genetic abnormalities? Well, as I said before, everything began back in 2012 with the creation of the cytogenetics lab at Clean Cells. The first important milestone was to be able to validate the assays in compliance with the GMP guidelines, which we achieved in 2015. This permitted us to work with many key players who were developing cell-based medicinal products. We can now say that we were fortunate enough to be there from the beginning, almost 6 years ago, and accompany our clients' products to meet the market, but at the same time, establish an industry standard for the Karyology and the FISH assays. As you can see in the graph on the right, there is a growing demand in Karyology these days, since there is also an increasing number of cell-based medicinal products. There is an exhausting list on this type of products on the slide, but before they are taken into clinical trials or reach the market, they must comply with the health authorities' regulation. And this is where we can support you. As you are probably aware, there is always a concept of risk analysis on the responsibility of the sponsor or the client. A risk analysis on the presence of genetic abnormalities is very relevant in the case of a cell-based medicinal product, as when present, they must be detected according to the many pertinent regulatory texts. However, the texts are not really exhaustive in the method and approach necessary to reach this target, and it is important for a biotech company to be supported by an experienced provider. And we will go into some of these details later today. We just have a quick slide here just to show you that it is evident that cell-based medicinal products took a quite favorable turn lately. with many different products getting clearance in the last few years. We can even quote, as you see, some products who reach the market like Yescarta, Kimbria, Tecartus, among others.
Okay, I think what is important not to say, Vasilis, is for our clients or people working at least on those type of products, what could be the cases, what could be the things that will trigger the need for this genetic stability investigation?
This is actually a very important question, Aymeric, and we can say that the cytogenetic assays can be used to meet the demands of the regulatory authorities for the evaluation of both the genetic identity but also the stability of a cell-based medicinal product. This evaluation includes the detection of cell contamination, specific cytogenetic markers during production, the validation of diploidy for human cells, and of course, the potential genetic instability of cell substrates and modified cells during extended cell culture, Aymeric.
You mentioned a lot the cytogenetic word earlier. Can you give a more precise definition for it?
Sure, Aymeric. It is true that many people are quite familiar with molecular biology, especially nowadays with the PCR testing for during the COVID pandemic, but not so many with cytogenetics. Very simply put, cytogenetics is a branch of genetics, of course, which studies the chromosomes on the cellular level. The classical cytogenetics approach includes a Karyology assay that we're going to talk about today quite extensively, while the molecular cytogenetics approach combines the power of molecular biology and cytogenetics in methods such as FISH, which will be also discussed later today. Well, Karyology is a very well-established method, and this is the first assay you need to consider when it comes to searching for genetic aberrations. It is a whole genome analytical tool which can detect very reliably all types of aberrations, including balance translocation and inversions.
Yeah, I think we got uh, what Karyology can bring, but can you explain in details the scientific aspect of these assays and explain to us how it works exactly?
With pleasure. Well, the key parameter here to be able to perform a Karyology assay is the presence of proliferating cells. During cell division, Aymeric, there is a specific step which is called the metaphase step, where all the DNA content of a cell is packed into clearly visible chromosomes. In order to analyze these chromosomes, we need to block the cell into the metaphase step using a specific chemical. In this case, we use colsamide. Then, using specialized equipment, we can capture really several metaphase images, and using the ISCN international guidelines for cytogeneticists, a specialized operator can classify the chromosomes into groups, establishing ultimately the karyotype of the cell. Finally, as you can see on the bottom left, there is a specific gene binding profile on the chromosomes, which will detect any potential genetic aberration, Aymeric. Yes, normally there should be a slide here for the ISCN textbook. Well, we just mentioned this ISCN textbook, which includes the international guidelines that set the rules for the description of a Karyology analysis result in a standardized way that can be recognizable by any cytogeneticist around the world. In addition, and very importantly, it defines clonality during analysis, which simply put is what renders a karyotype either diploid or positive for a given clonal chromosomal aberration.
Okay, Vasileios, you made it clear about Karyology. There's also the fish analysis. It will enable us to go deeper into the analysis and to understand what has been going on in the cells about the genetic rearrangement more precisely. Can you elaborate on this assay as well?
Indeed, Aymeric, FISH is a rapid and technically not very demanding assay that can detect, the target genes of any specific aberration based on the type of fluorescent probe that it is used. The big benefit of the method is that it does not require any proliferating cells, since the standard FISH analysis is performed just on nuclei. On the other hand, however, FISH is not a whole genome analytical tool, and the targets of the analysis must be predetermined. Therefore, in combination with Karyology, they provide a strong combinatorial kind of approach for the analysis of the genome. Now, on a more technical note, a standard FISH experiment requires the careful design of a double-stranded DNA probe, which is specific to a gene or a chromosomal target. The probe is labeled by fluorochromes, which are then detectable using an appropriate microscope. Several slides are prepared carrying the sample nuclei, and then using again specialized equipment, the target DNA and the probe are denatured simultaneously under specific experimental conditions, of course. And finally, the probe hybridizes to the target DNA. After a series of post-hybridization washes to remove the excess of the probe and, of course, unwanted background noise, hundreds of different nuclei images, like the one presented in the slide, are captured and analyzed by the trained operator for the detection both the number and the type of signals present in the sample nuclei. Back to you.
All right, thank you, Vasileios. That was very complete scientific explanation. I think our audience knows everything about the Karyology, the difference between Karyology, cytogenetic, and fish analysis. There's going to be an exam at the end of the session. I'm joking. All right, so this is the first Q&A session here. So I have a question just now for you, Vasileios. So it's about the specificity of the fish probe. So how does the fish probe works, and how can you know its specificity?
Well, Aymeric, each probe is carefully designed in advance to be specific to the target gene or chromosomal target and no other location in the genome. Then, after a specific set of experiments combining Karyology and FISH using this probe, we are able to prove and validate its specificity, but also that it performs exactly as expected, Aymeric.
Okay. Another question. So it's about the actively proliferating cells. So the proliferating cells, what does it mean in terms of criteria? I know this is really important for the karyotyping assay. Can you tell us more about that?
Well, simply put, Aymeric, actively proliferating, it means that we have happy and healthy cells in division. As you can understand intuitively, the more cells we have in division, the more cells we can block in the metaphase step. And of course, the metaphase yield is increased proportionally.
Yeah, and we want happy cells. That's something I like also. So another question here about the clonality in Karyology. So can you tell us why the clonality is important to measure when we are doing a karyotype?
Sure. Clonality actually is a critical parameter for any cytogenetic analysis, which indicates the existence of a clonal population of cells, carrying, of course, the abnormal karyotype, within, however, the normal or healthy cell population. The outcome of this process is what is considered genetic instability and detected using cytogenetic assays, Aymeric.
Okay, you have to spot the black sheep in the herd. Okay. So there's no more questions. No. Okay. So we should move then on the actual case study. So yeah, like I said, the case study are purely fictional, but they're really representative of what could happen during such genetic stability investigation for a cell therapeutic product. So the first one is about CAR T cells and is about the story of balanced translocation. So it's you know how it works with CAR T. You have this ex vivo autologous approach, and while you modify the receptor of a lymphocyte, and you end up with what you call a chimeric receptor, which is abbreviated in a CAR T denomination. So so Vasileios, can you tell us more about this project and what has been going on with this specific translocation event?
Exactly, Aymeric. Well, during an IND filing, a client needed to assess the genetic stability of these CAR T cells following the modification of two specific chromosomal loci involving CDA and CDB genes. During the preclinical development of the product, we performed a first Karyology analysis, and we were actually surprised, both of us, to detect a clonal balance translocation. Interestingly enough, the translocation involved the chromosomal loci, which were the targets of the modification by our client, Aymeric.
Yes, and once you got those results, those unexpected results, of course, what was the next step to really understand what was going on?
Well, the first thing we proposed was to perform a Karyology analysis to exclude the predisposition of the aberration in the donor cells prior to the modification. Even though it was quite apparent that the aberration was related to the modification, we had to be absolutely sure using, of course, our cytogenetic tools, Aymeric.
Right. So at this point, you could say with proofs that the genetic instability was due to the genome manipulation of the cells in order to change the receptor of the cell.
Indeed, the tested unmodified donor cells were negative for the aberration, and hence it was quite clear that it was the result of the cell modification.
All right, so next step would be to go deeper, I guess, with using the fish analysis with the fluorescent probe to really know the gene involved in this translocation event and know more about the gene rearrangement itself.
At that point, we already knew the type and chromosomal location of the aberration using Karyology. Then, using the FISH assay, we were also able to identify the target genes of the translocation, which were in fact the CDA and the CDB genes, as actually we expected them to be, Aymeric.
All right, so at this point, you made it clear for the client what kind of genetic rearrangement occurred and what gene were involved. Finally, what is the outcome for the client and how can it benefit from those findings?
The most obvious one is that we managed to detect and well characterize an unexpected aberration during the preclinical phase of the product, and of course not later. This triggered internal testing on the side of the sponsor, including, of course, many different assays that managed to prove to the authorities that this aberration did not give any tumorigenic or other adverse effect on the cells. And finally, after quite a bit of work and collaboration with our client, Aymeric, they managed to get a clearance and they moved into clinical trials.
All right, so the moral of the story here is always to go further when you have a bad finding to demonstrate to the agency that you have control and knowledge over your product to lift any concerns or blocking points.
Exactly.
All right. Now let's move into the case study number 2. This one is a story of a common genetic abnormality occurring in stem cells product that can turn eventually into a hurdle and make some difficulties.
In this case, Aymeric, a client approached us for the cytogenetic analysis of a human embryonic stem cell derived retinal pigment epithelium cells, or RPE, cell therapy product. And it is well known for quite a few years now that pluripotent stem cells are susceptible to acquiring chromosomal aberrations during extended cell culture in a phenomenon known as culture adaptation. Therefore, we propose to perform karyology analysis during many different production steps. The first karyology analysis of the cells was normal. However, further down the production scheme, we performed another karyology analysis, and this is when we first detected a clonal trisomy of chromosome 8.
Right? Are those genomic aberration like trisomy of the chromosome 8 common a common event in such cases, in such cell types?
Trisomy 8 is a less common but quite well-known aberration that has been detected during the extended culture of human embryonic stem cells. In fact, the presence of this clonal population carrying the aberration is in competition with a normal cell population, and we had to assess whether one is going to take over the other.
Okay, so the risk here would be to have a competitive advantage, right, of the abnormal cells that ends up overcoming normal cell population through time, right?
Exactly. Hence, after another karyology analysis on a later production step, we saw that not only the aberration was present, but it was detected in more karyotypes than before. This meant that the size of the aberrant clone was increasing.
Okay, and as usual, you went a step deeper with doing some fish analysis, but this time, what did it achieve?
Well, it was clear that the aberration was specific to these cells, and we used fish analysis as the fastest and easiest, actually, approach to further follow the size of the clone. And indeed, the fish results showed us that the clonal population was increasing even further. At that point, it became evident that the aberration was dominant, and we helped the client dodge a bullet, so to say, before applying for an IND. Back to you, Aymeric.
Well, thanks. We can now give it a conclusion to those case study and webinar. We made a short key takeaways point here, so you can have some points leaving us today, and then we will go into the second session of questions. I think we made it clear, and we noted today that Karyology remains the main tool in the regulatory context to address genetic instability using further cytogenetic tools. There are, as Vasileios mentioned, other approaches, more sensitive tools like the fish analysis we talked about to detect chromosomal aberration. But Karyology remains the first approach to really have a broader view and to understand what is going on. We can also say that since the analytical approach using cytogenetic assays is not clearly defined in the regulatory authorities, here at Clean Cells, we can use our extensive experience collaborating with key players in the field to really give you a support and give you, like I said, an industry gold standard to help you with the first development until the post-market authorization for those genetic abnormalities assays. So an important note before we end this presentation is to talk about our recent feedback from the agencies. So that's a very interesting point for the people developing such drugs now, that we had feedbacks from health authorities, and in fact, more often now, they want to know how many genetic abnormalities are within the product. They want to have a percentage of the overall cell population afflicted by the rearrangement. So to answer this new need, we are developing a full quantitative approach, complementary with karyotyping and fish analysis, which are more qualitative approach. And so the quantitative approach would be to use ddPCR method. So because it's still in development, we will not elaborate too much today about that, but we keep the door open and let you know that in the future, we will coming back to you with more feedbacks about that. And for example, there's a newsletter that will follow at the end of this year, talking more precisely about the ddPCR and what it can achieve for your product in order to really have a quantitative approach of those genomic aberration. So let's get into the question here. Of course, if you want to have some follow-up about this webinar, you can directly address a message to me. So you have my email address just on the bottom of this slide. Anyway, so I have a first question for you, Vasileios. So it is, how do you know that only the CD integrated genes, so CD, I mean, it's I guess it's for cluster differentiation, so it's like the receptor of the lymphocytes. So how do you know that only CD genes were translocated? Is there any other genes involved?
Well, actually, Aymeric, what we can say here is that by using FISH, which is a very precise method to detect the target gene of an aberration, if used properly, it can exclude any other potential targets. When used in combination with Karyology, they provide a very strong and complete and specific analytical approach.
All right. Well, I have another question here, a more really a question about how do you send the samples? So, I mean, I guess that the question is about sending corticell samples, or maybe cell-based product samples, maybe fresh cells or stem cells, all those kind of products. So how do you ship them to us, Vasileios?
Well, we can actually receive many different types of samples. The first one, which is the most typical, they are frozen vials containing cells. So they are delivered to us in liquid nitrogen vapor, and they are stored at clean cells until we begin the analysis. The second type, it could be live cell cultures. This needs a very close collaboration with the client in terms of the seeding concentration, the transportation duration, etc. But it's quite simpler than it sounds. Usually they are transported in room temperature or in 37 degrees, but even room temperature can work. And after the reception at clean cells, they go immediately into cell culture in our incubators, and we continue with the karyotype analysis. We have also some not so common cases that a few of the sponsors, they prefer to fix already the cells and provide us a fixed cell suspension in order to begin the karyotype analysis. This is definitely possible. It has worked in the past, but again, we need to make sure that their fixation protocol and the duration of the cold semi treatment, etc., there are different variables in the protocol. They are optimal to give us a good metaphase yield and with good quality metaphases. So just to summarize, we can receive any type of sample. In some cases, we need to talk a little bit more before reception. In the frozen vials, it's quite easy.
What do you prefer?
They are all the same to me. No, but honestly, the simplest one would be the frozen vials because they can remain in storage until we make sure that we have our planning ready and everything goes into place. So the safest and easiest to handle would be the frozen vial. The most tricky one would be the live cell cultures because we need to be in place and follow step by step immediately the process. And the fixed cell suspension, it's also, as long as it works, Aymeric, it's a very safe approach because fixed cells are very stable and quite resistant in transportation, many other factors.
Okay, you made it clear. I have another question, one last question exactly. So per the regulation, it is mandatory for my CHO produced cell line. So sorry, is it mandatory for my CHO produced cell line to have a Karyology assay? Can I use other assays to achieve the same compliant results?
Well, there is a very simple answer to your question, Aymeric. If we use the European Pharmacopoeia chapter, most of you probably know about it, the 5.2.4 as our reference, any CHO produced cell line needs to be tested for any specific cytogenetic markers and the number of chromosomes at minimum 2 different production levels. Usually it can be the cell seed or the master cell bank, and definitely the end of production cells. More generally speaking, in some cases, and really I'm not talking about only CHO cells here, there are other assays that can lead to the same conclusions, but none of them, for the moment, can substitute Karyology as a complete whole genome analytical tool in GMP conditions.
Yeah, Karyology is the way to go. So I guess it concludes our webinar for today. I hope you had a good time with us and that you learned some things today. Thank you again for attending this and share this time with us today. And thank you, Vasileios, for all your kindness and time explaining your job and may I say it, your passion.
Thank you also, Aymeric, very much for the invitation and coordination today. Thank you, everybody that attended. Please do not hesitate to contact Aymeric, and we can discuss further if you have any more questions, or we can definitely solve the almost all kind of type of questions. Thank you. Have a lovely evening. Bye-bye.